PCR importances. PCR used to make millions of copies of a target piece of DNA. PCR allows specific target species to be identified and quantified, even when very low numbers exist. PCR important for research purposes, and PCR was first used to analyze the presence of genetic disease mutation. PCR can be used to produce huge amount of pure DNA samples from a limited sources. Detection of genetic variation. Genetic variation can be expressed by variations in the ploidy levels, by structural changes in chromosome, by the loss of one or several chromosomes, or by mutation at the gene level. Alteration in ploidy level can be detected by karyotypic analysis of metaphase chromosomes. Chromosomal changes and DNA variation do not always reveal alteration in individual genes. A precise determination of changes in a particular gene sequence resulting from tissue culture can obtain by restriction of fragment length polymorphism (RFLP). The RFLP analysis, the random amplified polymorphic DNA (RAPD) analysis. Factors of genetic variation: gene mutation, recombination, selection, and gene flow. Gene mutation, alter of nucleotide bases by natural process or deletion, recombination, by independent assortment between chromatids of homologous chromosome during meiosis, selection, preference phenotype, gene flow, gene transfer from two different populations. Mutation to change their protein function. A missense mutation is a mistake in the DNA which results in the wrong amino acids being incorporated into a protein. A single DNA sequence change results in a different amino acid codon which the ribosome recognizes. Changes in amino acid can be very important in the function of a protein, but sometimes they make no difference at all, or very little difference. Missense mutation cause an amino acid to be incorporated, which makes the protein more effective in doing its job. It causes the protein to be less effective in doing its job. Mutation importance in plant biotechnology. New varieties produced in mutation breeding, a useful way of making a one-point improvement of a trait in original cultivars. Mutation breeding can change a dominant trait to a recessive one mostly. Genetic transformation produces an additive one-point improvement, where mutation breeding produces a subtractive one-point improvement. Genetic transformation can modify target traits by direct incorporation of related genes. Genetic change has provided the natural variation for the species evolution that important for adaptation to the natural environment. Process of species domestication and crop improvement, increasing crop yields in stages of crop evolution. Domestication yields from plants growing in the wild were seasonal, sporadic, and totally dependent upon the prevailing natural environment. Yields are varied unpredictably, bringing years of feast and famine. Domestication represented a major event in human and crop evolution as it provided a degree of food service.